Hello everyone. Welcome to the Input Modules video tutorial. My name is Pearl Ryder. I am a postdoc in the Simony lab at the Broad Institute, which is part of the imaging platform there. And today I'm going to give you an overview of the different image import modules, and then my colleagues will demonstrate how to actually use these modules for different types of data: two-dimensional, three-dimensional, Z-stack, time-lapse data, etc. Okay, so considering the different input modules, within the CellProfiler window, these modules are located at the top left of the window. And we have the images module, metadata, names and types, and the groups modules. Considering them in turn, we'll start with the images module. This is where you can let CellProfiler know where to find images on your computer to analyze. You can drag and drop folders of images here onto this panel, and then you can apply filters to those images. So for example, if your file folders contained both image files and other types of files, you could apply a filter so that you were only trying to analyze the images within CellProfiler. One thing we recommend when you're actually naming your folders and your file names is to avoid any special characters such as parentheses or periods. And this can help avoid any errors within CellProfiler when it's trying to parse and read those file names. A common question that we get is, how do I know what types of images CellProfiler can analyze? To answer that question, you can click on this little question mark when the images module is highlighted. This question mark button will bring up the help manual, and that help manual has a lot of information about the images module, including that it can read a variety of image formats, and that you can follow this link to find a website that will tell you exactly what types of images CellProfiler can analyze. Once you've configured the images module, the next module is the metadata module. Now, this is an optional module that allows you to extract lots of different experimental conditions for each image, as well as different channel information about which image that corresponds to. So for example, if your experiment had both channel 1 and channel 2 for each field of view, you could extract that information using the metadata module. This metadata module can extract from the file and folder names, or it can import from one single file, or it can also extract from the image file headers. So you have a lot of different options for the metadata module. These different conditions that you extract here can be used within the names and types and the groups modules to configure those. So sometimes if you're getting an error within the names and types module, it's actually coming because of the metadata module. So you might need to check that out. Okay, next up is the names and types module. This module is where you actually give a name to your images that you will then use in your CellProfiler pipeline as you build it. And it's also where you can configure image sets. So we can take a look at the help to get a little more information about what an image set is. And this gives a nice example where it shows that you can have, for one field of view, you can have multiple channels that were imaged, such as this nuclear channel and this cytoplasmic channel or GFP signal. Within names and types, what you would do is you would configure so that each field of view that has two matching channels that go along with it are configured into one image set. So in this example, an image set would have a nuclear image as well as a cytoplasmic image. Finally, the groups module is another optional module, and this is where you can group together images that are all within the same field of view. So for example, if you have Z-stack data, in Z-stack data you will have multiple different planes of image sets that are all corresponding to one field of view. And in the groups module, you could group all of those Z-stacks into one group per Z-stack. That would then allow you to make a projection, a Z projection, for example. Alternatively, you could have time-lapse data where you group each time-lapse movie into one group, and then you analyze that group using the CellProfiler pipeline that you build. So that's an overview of the 4 different input modules for CellProfiler. Up next, one of my colleagues will be demonstrating how to configure these for two-dimensional data.
Hi everyone, my name's David Sterling and I'm a postdoc in the Lab. And today I'm going to be running you through how to set up a simple 2D experiment with cell profilers for input modules. So to do this today, I've started with the example human dataset available from the Cell Profiler examples repo. And we have our input module here in Cell Profiler, and you can see a big target here for dropping files and folders in. So to start off, I'm just going to drag my images into that, and you can see it's now populated the file list. Now, one of the things that you can do with this module is you can actually double click on an image to preview it. So we can see here that one of our images is some nuclei stained with DAPI, and one of the things you may have already noticed is that we actually have 3 different channels here rather than 3 image sets. So we have channel 0 here with the DAPI, but we also have channel 1 with a slightly different stain, and channel 2, which again looks quite different. And one of the key goals in the 4 input modules is to try and change a long list of files into a series of sets of images matched to each other. So in this case, rather than having 3 sets of images to run, we actually have a single set with 3 channels. And that's what we really need to use the rest of the input modules for. Now, we're very fortunate here in that we can tell which channel the image is just from the file name. If we couldn't, there are several different ways that we could put out more detail about the images using the metadata module, but that's something that will be covered in a later tutorial. For now, we're just going to go straight to our NamesAndTypes module. Now you can see by default, it will assign the same name to all images. In this case, it's using DNA as the name. If I were to update, my preview here is showing me that I have 3 image sets with 1 image each. Now, obviously, we have these 3 channels, so we actually want to make 3 channels per image set. So I'm going to switch this to images matching rules mode. Now, if you remember, our channels are determined by the last few characters in the name. And you can see here we have a series of building blocks to basically create a rule which identifies a single channel. Our first channel is still going to be DNA, so we'll stick with that name. We do have different options here, so we could use the folder name or the extension and all sorts of things here. But we're just going to use the file name at the moment, and we're going to say, yes, it does contain, and we're going to say it's going to be d0.tiff, because that should be our first channel there. If I were to update now, you can see it's only now got the 1 image, and these other 2, D1 and D2, aren't matching to that rule, and so don't have anything assigned to them. In order to make this work properly, I'm going to need to add additional rules to pick up those other channels. So if we add another image, we would now do d1.tiff, and maybe give that a different name. GFP is fine. I can also duplicate the same settings if I want to do a very similar set of rules. So d2.diff, we could call that one RFP. Now if I update, what you see is you've got these 3 channels that I've named and the 3 images correctly assigned to them. What we can do here, if we wanted to do something more fancy, would be to add multiple different conditions. So we can say, maybe we also want to make sure that the extension is a proper image file. At the moment, all of these rules have to match to put something into that D2 list, or the RFP channel. We have the option to also choose whether it matches a single one or multiple different rules or any other list of rules. The important thing here is that it's customizable and you build the rules to match to a particular image channel that you're wanting. And the way you can imagine this is that when you run this module, it will basically divide your images into however many of these different categories that you created and then match them up accordingly. By default, it will then pair them in the order that they're generated in. In one of the later tutorials in this series, we'll be covering how to pair instead with metadata and do more complicated matching in case your images haven't all been brought in the correct order for each channel. So just to confirm, if I update here, you can see I've now got my 3 channels available in a single image set. If I wanted to now, I could quite happily use something like the threshold module, and you can see that I have those 3 channels available to me. Indeed, if we threshold the DNA and get into test mode, you can see that's all working fine, and we've made a nice segmentation start. So for most experiments, you'll be bringing in your images typically in this kind of single plane per file grayscale format. But there are other scenarios where you might not have grayscale images coming in. So let's take a look at some of those. So I have here a series of different images. Well, it's actually the same image, but stored in different ways. Right here I have a PNG file which stores all 3 channels overlaid in red, green, and blue. And this is quite a typical thing to find when you've got microscopes or overlays. To handle this sort of thing in CellProfiler, it doesn't really matter what the format is. CellProfiler can read most common formats and a lot of the more obscure ones. But I'm just going to use the PNG for now. And you can see that when I open this up, CellProfiler has automatically seen that it's a color image, and it still displays just fine. However, this is basically merged data. The 3 channels are all part of the same image object. And what we need to do if we want to work with that is split it up. Now, with a file like a PNG or sometimes a TIFF, if we go into our names and types, we no longer have these 3 different wavelengths. We can't just say, give me that, let's say, hey, there's no image sets here. We're fortunate that we can just use all images mode here because we have one image, which will be one set, and update. Now, while this is assigned correctly, this is not a grayscale image, and we won't be able to work with it properly if we declare it as a grayscale image. So we need to make sure we select the color image option here to tell CellProfiler to expect more than one color in this particular image. And this is important because a color image will not work with, say, the threshold module. If I were to try and run this now, it will probably pop up an error. Yeah, it wants a grayscale image. So there are several different ways we can address this within CellProfiler. The easiest one for this scenario would be to use the color to gray module, which is right here. And this module has a few different modes for taking a color image and turning it into a gray image or images. The default method is to combine the 3 channels into a grayscale image. I'll just show you how that looks right now. You can see it's taken our 3 channels and turned them all into gray, overlapping them with each other. That image can then be used as normal in any of the other modules. The other mode here will be the split mode, where we would take each of the color components and put them out into an individual image. If we run it this way, our single color image goes into those 3 grayscale channels, and those are a lot like the 3 images we had in the original set where they were grayscale. So we could then operate on these just as normal. Now there are some other image formats which may display as color. If I were to bring in this image here, this is called a composite. And a composite is similar to a color image, but the individual channels aren't actually stored as the same wavelength. Now, CellProfiler by default has been told this is a color image, but if I were to open this in something like ImageJ, what you'd see is that there are actually 3 different wavelengths. And ImageJ, again, is showing them in color, but I think Windows Photo Viewer, for example, that's a bit better. We can see that these are actually individual, what's called planes in the image. So to put this another way, the composite image is a similar data type, but instead of having the 3 colors squashed into a single object, you actually just have a file with 3 grayscale images stacked next to each other. Now in CellProfiler, we could declare this as a color image and use color to gray again. Color to gray does actually have a channels mode where it can pull out individual channels. And that would be geared towards this kind of composite scenario. One of the important things about the composite mode of storage is that you can actually have more than just the conventional red, green, and blue channels. You can have as many channels as you want in that image, so you may need to take out many individual channels. Now, there is another way we can handle this though. So if we go back into the metadata module, one of the options we have here, instead of extracting from folder names, we can actually extract from image file headers. And that means look at the file and tell me what's in it. By default, the image is just seen as a single plane or image. If I were to extract the metadata from this image and then update, you can see it's actually been able to detect that there are 3 images within the one file. And this is quite common with a lot of the proprietary microscopy formats. You can see that what we actually have here is C for channel, channel 0, channel 1, and channel 2. And many formats such as CGI or ND2 will contain not just multiple channels for each plane, but also multiple frames in each series. So there are also T for time and Z for Z-stack parameters here, and you may indeed find that your images will have multiple time points or Z planes in the same file. So it's this extract metadata from image file headers option here, which allows you to pull those out. However, this only works with a composite kind of format. It won't work with a proper overlaid RGB image. So we've already come up with our rules to identify 3 individual channels earlier based on the file name. What we'd do to get this one working with the composite image would be to, instead of using the file name, use the metadata. So we have access to all the metadata, which is extracted in the metadata module. If we were to say, I don't know, C0 there, we would then be able to do the same thing for the other 2 channels and hopefully get a full image set. Let's try that. So again, we're going to match metadata, channel 2, the update. There we go. We've got our image set. What you'll notice is that all 3 of these planes are coming from the same original file. So that's how we split up a single file into multiple planes if they're contained within a single container format. Now there's one more scenario I would like to show here, and that would be if you're working with an image like this one here. Now this is a bright field image which has been stained by immunohistochemistry, and again, we can open this in CellProfiler just fine, but this isn't really a channel-based stain. There's not like a red, a green, and a blue component operating individually. They're all mixed together because it's actually just a raw color image. And the color to gray module wouldn't necessarily split that very well. I'm just going to set that up as we had before. There we go, we'll call it a color image, and we're going to color to gray. Now, if I was to try and split the red, green, and blue components from here, what you'll see is that it doesn't really do a very good job. You can see that there's red, green, and blue components to the actual stain we're looking at. So when we want to do something a little bit more complex like this, we have another module called unmixed colors. And this is designed a little bit differently in that it's built around trying to take a common IHC stain and pulling out a particular color channel into its own fluorescence marker. So you can see we have a bunch of common stains that you might hear, and I guess hematoxylin is relatively close to what we're trying with this bright field image. If I were to run this, what it will do is take the original image and pull out a very specific wavelength as a black and white grayscale image, similar to the other kinds of images that we've seen. So you would be able to use this to perhaps do your segmentation just like you would with a normal fluorescence image. One of the things to mention about this module is that we also do have a custom function here, where you can set a individual color to your particular choosing that you want to pull out. And you may not know the absorbance automatically, so there's also an estimate tool. And the idea here being that you would supply an image which just contains one of the areas of staining that you want. So in this case, we might zoom in and crop out just this region here, which would be a good example of the color we wanted to pick out. And CellProfiler could estimate a new stain identity from that. You can also pull out multiple stains if you have, let's say, 2 or 3 or 4 stains in your histology specimen. So that about sums up the basics of doing 2D setups in the input modules. There are some additional options which will be covered in other tutorials, but yeah, the important thing to do is to keep an eye on this pairing window here in names and types and make sure that you've properly associated your individual channels, if you've got more than one wavelength, into proper image sets which match and pair correctly. Thank you very much for listening. I hope this was helpful.
Hi, I'm Erin. I am a staff scientist in the Simony Lab in the imaging platform of the Broad Institute of Harvard and MIT. And today I'm going to be showing you how to use the input modules in CellProfiler to input data sets that have images that are shared across the data you want to analyze. So I'll show you two examples of this. One is a data set that uses illumination correction. And in this example, we have a different illumination correction that we want to apply to each channel, but it needs to apply to every image within that channel. And the other example is a data set where we have a mask, and we want to apply that mask to every single image in the data set. So to get started, the first data set that we're going to use, so I'm going to start with just the basic 2D image set example here, and then build upon that, just as David showed you in the last video. Here I'm starting with just dragging and dropping some images into my input images module here. And then I am going to extract some metadata from them. So first, what I want to do is, for these, I want to extract what channel they are in, because again, I'm going to be applying an illumination correction on a per channel basis. So I want to make sure for each image that I know what channel it's in. And so for these, the channel here is right in the channel name, channel space, and then either 1 or 2. And so what I can do is use this regex here and have it automatically figure out what the channel is. And so the way I am doing that here is I'm inputting channel and then a space. So that knows it's going to search, find channel in the space, and then it looks for the channel. And I'm telling it that channel is the first single number that it finds between 0 and 9. And so here, in this test text that it's taking from this this example image in our data set, this regex successfully finds channel 1. And so if I do this and then update, it has successfully figured out that these images are from channel 1 and these images are from channel 2. However, we are going to need an additional piece of metadata in these to be able to distinguish between these two images. One of them is, they're coming from separate wells or separate sites, and so I'm going to add in an extra bit of information and also tell it to extract, let's call it a site. And that is going to be the next two numbers that come after the dash here. And so now it has figured out that we have in our 4 images, there are 2 different channels, 1 and 2, and 2 different sites represented. And that means that it's found a single image for each site. It's found a single image in each channel. Great, so we have that metadata extracted. And then if we were to go into names and types and give them a name, because we need to have a name for these images when we're working with them in the various modules that we'll add to our pipeline, I want to assign a name, not the same name to all the images, but I'm going to set a rule here. And there are certainly different ways that we can do this. One example would to be file does contain channel 1, and then I will add another image here, and file does contain channel 2, and it's fine. We can assign them DNA and GFP, that's fine. So if we update here, it's great. It's found 2 sets of images. This one here from this well, this one here from this well. And the way that it matched them here is just matching them in the order that it found them, which is fine. Other ways that we can handle this, since we did extract metadata, we don't actually have to put in information about what's in the file name itself. We can actually match the metadata. And so here we can say metadata does have channel matching 1, and metadata does have channel matching 2, and we're able to get the same thing. So there's often different ways that you can handle assigning names to things. And so we're able to handle here the most basic set of images that we have in this data set. However, as I mentioned before, we want to be able to add illumination correction to these images in our pipeline. So I'm dragging and dropping in these additional illumination correction images. And so if I double click on them, I can open them and see this is what it looks like. It is actually a NumPy array. That's why it has the file ending NPY. But we can visualize it, and this is what it looks like. And so if we were to proceed from here, we would actually end up getting possible confusion and error, because currently we have this filter images turned to images only. And so if we show what that looks like by applying filters to the file list, it will then gray out any file that it doesn't recognize as matching those filters. And so it actually grays out our illumination correction, which we don't want. So instead, we can either say no filtering, because we have dragged and dropped in exactly the images that we wanted. Or we can set custom, and you might want to do this if you're just dragging in entire folders of things, but it turns out that you have, other files in those folders like text or a README or, whatever, that you don't actually want CellProfiler to recognize. And so one setup that we could use here, so we want to match any of the following rules where the extension is an image file, so it's telling it, yes, we want all images. And then perhaps we'll tell it also file does contain .npy. And so if we apply that to the list, it recognizes all of our image files and our NPY files. So we can proceed. In metadata, you can see it's automatically updated, and this regex that we've set here doesn't find any channel or site information for these files. So we're going to need to add another extraction method to be able to get some information about these illumination correction images, because again, we need to know which channel they're in, because we're going to apply a single correction image here to every other image that is in that channel. And so we can actually perhaps start with some of the same regex here, but if you notice, it just so happens the way these are named, these have a space after channel, these do not. And so if I delete that space, it's then going to search for channel and say that the channel it finds is the next number after that. But I also want to make sure that it is not getting confused and finding that same string here in these 4 images. So I'm also then going to say right after that number, that is the channel number, then comes illum. And so if I update this now, it successfully found that this, this correction image is channel 1 and this one is channel 2. So we have it finding the metadata that we need to be able to proceed. Now coming to names and types. So doing it here by order, the problem is here, it's saying it's found all of the, just using what we have here, all of the DNA images are in this list, and all of the GFP images are in this list, which is true that the that this is a, this correction image is a channel 1 image and this is a channel 2 image. However, the way it's setting this up is essentially it's going to try and process now each of these rows, which are, a stack essentially of 2 channels, making up a single site is what we called it. And so here when it's parsing the metadata like this, it's going to try and process these NPY files just like it does in a pipeline, these other sets. So that's not actually what we want. What we want it to do is configure it so that for this image, it knows that this channel 1, site 1 is the DNA image, this channel 2, site 1 is the GFP image, and then we need to tell it that these illumination correction functions are part of that same set. But we also need to figure it out so that they can apply to all of the images that we're going to be going. So that's why, again, once you get this more complicated situation, you definitely don't want to just use order. We need to pass things by metadata. And so if we did that now, it's going to give us some errors. And the reason that happens is, again, we have yet to tell it how to handle these illumination correction images. So currently, metadata does have channel 1, assigns things to DNA, and channel 2 to GFP. But we need to get a little more explicit than that because, again, those illumination correction images, we already figured out that they are also channel 1 and channel 2, but we need something that will separate them here in the names and types. And so one way that we can easily do that is we know that the images that we want to process are TIFFs, and these corrections that we want to apply are NPY files. So we can add here another condition where file does contain TIFF and file does contain TIFF. And now when we do that, well, it's still going to give us a separate set of errors, but it is able to find, because it doesn't know how to handle the NPY files that we've put in but not found matching rules for in names and types. But it is now successfully able to figure out the DNA and the GFP. So now we need to add in rules that help it figure out what to do with the NPY files. And so in this instance, we can say file does contain NPY, and remember we also need to be able to parse it by channel. So metadata does have channel matching 1, and here we'll call that the illum for the DNA. And the image type here, we're no longer talking about a grayscale image, we're talking about an illumination function. And so this is a separate class that CellProfiler knows to handle differently than the images that make up the data set. And so we will add one more image here where, again, file does contain NPY, but also the metadata does have channel matching too. And this we can call the illum for the GFP. And again, I will set this to be an illumination function. And now, so we're getting close, if we click update, we still have errors because it doesn't know how to match up these different classes. So it's figured out that DNA, GFP, illumination for DNA and illumination for GFP are separate things, but it doesn't know how to match them. So what we want to say is that a single set here is that the DNA and the GFP have matching sites. And so we also need to make sure we've selected an option for the illumDNA and the illumGFP, and we need to explicitly tell it none, because the illum functions are not different depending on the site. We want them applied to every site. And so now if we click update, what we see is that our images have been correctly split into two different sets. We have them, the DNA channel, the GFP channel, and then what you can see is that the same illum function is going to be applied to every, we will end up telling it later in a module what to apply it to, but when we say use the illumDNA function, or illumDNA correction, it knows to look for the same file each time. And so putting that into practice, we can then add a module, and if we do a correct illuminate apply, we can say, okay, every time we have a DNA image, we're going to apply illumDNA, and every time we have a GFP image, we want to apply an illumGFP, and we can call this like corrected DNA and corrected GFP. And so if we enter our test mode and we run this, we can see what that looks like is that this, for this single set, so it picked the first image set in our list, and it took the DNA image, it applied the DNA illumination correction, and then this is what our output image looks like. And did the same thing with the GFP image in this set, applied the illumination function, and this is the output that we get here. And so that is how you handle one example of how you can handle illumination correction images across a data set. And so now I want to more quickly, because we've already been through that once, show you how we might handle another example using a mask. And so I will just open up a new copy of CellProfiler, so we don't have to worry about overwriting what we've got in there, and move on to another example. And so in this example, I have three images, three separate plates that look like this. And I have here this plate template. And so what I want to be able to do is take this template file, which is a binary mask, because it's either black or white, and I want to be able to apply this to every plate in this data set so that we know we are measuring the exact same region. And so in this instance, where we're counting yeast colonies on a plate, applying this mask ensures that we aren't accidentally measuring or getting confused by anything that lies outside of the plate, or the plate edge can complicate things. And so by masking that out, it makes it much easier to take measurements, to find objects of interest, and there's lots of different reasons you might want a mask applied to every image in your data set. And so to do that here, we are going to, again, extract some metadata. And in this instance, I see that all of our images are named, the images themselves start with name, and then they have plate. And so we'll just say plate is the number that comes after the 6 dash in our file names here. So if we do that and update it, we can see that it successfully found a plate name for 1, 2, and 3 here. And so that's great. That's all the information that we need here, because again, this mask, we don't actually want to assign a plate to it because we want to be able to apply it to every single plate. So if we go to names and types, we will set some names to images matching rules. And great thing about setting metadata is we can use it to make our rules. And so here we can have metadata does have plate, well, actually, here, rather than, since these are all in the same data set, let's just say file does contain 6-, and we'll say these are our sample plates. There we go. So if we were to do this, it's going to successfully find our sample plates here. But again, we want to be able to also apply this plate template, and we want the single plate template to apply to every single image in the set. So to do this, we want to add a single image, and here it will let us just browse for the path. Oops, if you double click, it will open up the image for you to see. And then, so you don't need to do that. It's just a single click and then an OK. And I'm going to assign this, the name I'm going to give it is my mask. Here, again, I want to set this as the image type is not a grayscale image. I want this to be a binary mask. And so now if I do this, what it's saying here, if I update this, it's finding 3 different image sets that it's going to go through. Sample plates 1, 2, and 3, which is great. And it's going to apply this same mask to every single sample plate. And so we can see that in action. If we go to add in a mask image module here, we can say the input image is going to be our sample plates, and the output image will be our masked plate, and the mask we want to use is an image, and it is our mask. And so if we enter test mode, and we can see what happens, here we have our sample plate, and once we apply the mask, it becomes then our masked plate. So that is how you use the same sort of logic to have a data set, handling a data set where you have single or few images that are applied to many other images across the data set. So thank you for watching this tutorial.
Hi, my name is Barbara, and I am a postdoc at the CMINI Lab. So I'm going to continue this names and types tutorial, and we're going to talk to you today about the special case using Timelapse data. So for this case, we have 3 different movies, and the TIFF frames for the TIFF files that correspond to each frame within the movie are localized on these 3 different folders. So first of all, we're going to select those folders and drop them into CellProfiler. Then we're going to use the metadata module to extract some information from those files that then we can use when we save our analysis. So first we're going to extract information from the file name. So we had 3 things on this file names. So we have the specimen, we'll have the stain, and we have the frame number. So we have this little, this one is the text, I can use this, and this is a number. Okay, so now you see we have the specimen in red, the stain in green, and the frame number in blue. And so if you need help with the regular expression architecture, you can always click on this question mark, and there is examples for you to follow so that you can get the information that you want out of your file names. And you can keep this in mind when naming your files when you're acquiring your images. So we're going to add another extraction method, and it's named from the folder names. So we're gonna get the run. And we're gonna skip some of the folders at the beginning of this path. Okay, so when we have the name of the last folder that we have, that corresponds to the run. So we're going to hit update, and then you see this table then populates with information for both the frame number, the run, the specimen, and the stain. So then we can use, we can either, in this case we only have one type of images, so we can just give the same name to all the images, or we can have the images that match some type of rule. In this case, for example, the metadata does have a stain matching GFP histone, and this is the GFP image. So you can update this, and you see we have all the GFP files here, in this case 63, but we want them to separate each one by the different movies they belong to. So then we can use the groups module and use that run metadata that we extracted, and now you see that they are grouped according to the folder name, and you have 21 frames for each one of the movies. So for a different, in a different case where you have a movie where you have one of the images, either you have a mask of the objects that you want to measure, or you have one image you want to use all throughout the movie to either like mask a well or some or other examples of that, then we'll have we can use this input modules to tell CellProfiler, okay, I want to use this image on all the frames of the movie. So in this case, I'm going to select this other movie folder and drop it into CellProfiler. Then the metadata structure in this case is the same, so we can just update that. In this case, we have just 21 frames and an extra type of stain, which in this case is the nuclei. So on the names and types, we can add another image. And in this case, the metadata have stain matching the nuclei. For our case, it is not an image, but it's an object. And we can call it the nucleus. And so for CellProfiler to put that image on all the different frames of the GFP image, then we need to have this image set matching method using our metadata. So first of all, we can match them by run in case we had like more than one movie. And then we can match them with the frame number. So in this case, GFP will have a frame number and the nucleus has none. So when we update it, you see that for the GFP, we have all the different frames. And for the nucleus, we only have that one frame that we have, but it is on each one of the pairs. I hope this helped you, and there'll be one more of our team members talking to you about 3D images in CellProfiler. Thanks.
Hello everyone, I'm Nasim Jamali. I'm a postdoc in the imaging platform in Lab. And to continue what my colleagues have shown you so far, to configure the input modules and how to feed different types of images into the CellProfiler, I'm going to show you how to adjust the setting if you have Z-stack and multi-channel images, and what to do if you want to have Z-projected images, and how to feed 3D images into the CellProfiler. So I have a copy of CellProfiler open. I'm gonna load a couple of images that these are merged Z-stack. If I open one of these images in Fiji, I can see that they have 2 channels and 3 Z-planes. So I'm gonna pass both of these images as like all the images, and when I extract metadata, if I go ahead and extract from images file headers, hit extract metadata, and when I update, we can see that we have the 2 channels, and here we have the 3 zip lanes. I'm going to keep it simple and not add another extraction method and just move on to the names and types. And here I just want to pass all the images without having any matching rules, basically. So if I pass all of the images, we can see that we do have 12 image sets here. This is one way to go about this type of images. And then I want to make sure that in grouping, I separate based on the channels, and I need an identifier to basically be able to separate between the file names. And here my file location, for example, shows me that I can separate based on the names using these file location. So in groups, if I want to separate them based on the channel, I see that I have 2, and then I want to add another method, which is another metadata category, which would be my file location. So now I see that I have 2 images, and each of them have 3 zip lanes. Depending on the type of image analysis, we sometimes make maximum projected images from the Z stacks for each individual channel. And we use those images for segmentation. So here, if I want to create a Z projected image, I add the make projection module to the pipeline, and I definitely need save image module. So then I'm going to go ahead and here say that I'm going to load my images and have the maximum, and we can name it anything. We can say max projected image, for example. And in save, I want to make sure that I'm saving these projected images. And I want to get it from the image, the name from the image file name. And from the file name image, I want to add a suffix to be able to separate these based on the channels. And if I right click here, I can separate based on the grouping that we identified before, we defined before, basically. And I want to save it, and I want to make sure to save it in the last cycle. So now, because in the grouping I've separated based on the channels, my outcome would be something like each of the images with individual channels saved separately. There are other ways to do this as well. If your images are not already merged, you could separate them by channel in names and types, and then have a projection, make projection module for each of those individual channels. Now, what to do if you have multi-channel Z-stack CZI files? For these, I want to load these 2 CZI files that I have, and then filter images based on image only, go about configuring the metadata. So I want to extract from image file headers, and when I extract metadata, if I just click update here, I can see that I have 3 channels, and I have different zip lanes. I want to just add another extraction method based on the file name I want to be able to separate those. So I want to keep it simple because I only have 2 images. I can just take the entire file name as a image name or file name, and I want to make it simple and just call it file name. And when I update, we can see that another column added. So I have an identifier to be able to separate these based on file name. We could again use the file location like before in the previous example as well. And then in names and types, I see that I have 4 channels. So I have 4 channels and I want to just name them and define new rules. And as you saw before, metadata have channel matching. If it's 0, I want to call it channel 1. I could just duplicate this, channel 1, and do the same for all channels. And then I have 3, I'm going to call it channel 4, and then set the matching based on order. When I update, we can see that we have 111 image sets, and before in metadata, we had 444 because we had 1 row for every individual channel. But now that we created an image set, we do have 111 image sets. And then in grouping, I want to make sure to separate them based on the file name that we defined. And when I do that, I see that my first image has 55 zip lanes and the second one has 56. So it is okay that our images have different zip lanes. Now, what I want to do is to save individual channels for each of them have a different Z stack. So I want to add save images modules. Now I want to save individual channels and the Z stack for each of them. So I go to save images, and in here I want to make sure that I'm saving as a movie or a stack, and I'll go about adding the information for my first channel. And yes, I do want to add a suffix to be able to separate those. And here we want to make sure that we are saving based on Z slices. And I want to do this for all of my channels. Channel 2, similar, and then duplicate for channel 3. This is a bit dangerous, just when you do this type of just duplicating, we want to make sure that you change the names as well. So the outcome here would be 1 Z stack for each channel, which these can be then used in a different pipeline in CellProfiler for 3D segmentation. And if I open one of these Z stacks for each channel, I can see that it's a series of images saved for each channel. Okay. The last part I want to show you is how to load these Z stacks for 3D analysis. Now, if I go ahead and load the output of what we just created in a new copy of CellProfiler, then we want to see how to basically load the 3D images here. So I want to extract metadata from image file header. And when I would extract metadata and update, we can see that we have multiple zip lines. I want to be able to separate based on file name as well. So again, keeping it simple and take the entire file name as a method of extraction. And again, when I update, I see the file names for each of the channels. Then in names and types, I want to make sure that I do have the process as 3D selected. And I want to have image matching rules. So if you're wondering how to find the relative pixel spacing, we can, if you load one of the images and go to image show info, we can see and look for the voxel size. We can see that it's 1 by 1 by 1 pixel. So here we add 1 here. There are examples that this is not only 1, and it may be more or less. And then I want to go ahead and make sure that we define and have some matching rules. And my channel one, just to make it simple, I'm just gonna call it, if my file does contain channel 1, call it channel 1, and so on for each of the channels. Channel 2, and then I just duplicate all of these. Channel 3. And channel 4. And I want to keep it based on order. Again, we see that from 444 rows that we had in names and types, now we only have 111 image sets. Then I can go ahead and add modules for 3D segmentation. We want to thank you for listening and watching this video. We hope that you find this helpful. And if you have any questions, please ask on the image.sc forum. Till next video, goodbye for now.